Okay, the next speaker is Greg Carter.
Yeah, thanks, and thanks for this wonderful meeting. I've really enjoyed it so far. And I'm a mouse person at the moment, but I have worked in yeast genetics, so if you put me in the right environment, you'll get some cryptic yeast genetics from me. So I guess we're continuing with yeast at some level. But I'm going to talk about a mouse cross, and we're looking at genetic interactions on a very large scale for some body composition traits. And so what my lab does is to try to look across patterns of epistasis in cross populations, for the most part, populations of mice, and interpret those cases of epistasis, statistical epistasis, into models that might be something like this here, where we have a couple of variants or a couple of genes that might suppress one another and in turn have an effect on a phenotype. And in this talk, I'm going to particularly ask, when we do this at a large scale, what can these models tell us about the genetic architecture of these complex traits? Okay, so this is my one slide for the method. We've done a lot with this method, but we call it CAPE. It's a combined analysis of pleiotropy and epistasis. And the basic idea here is that we want to switch from having epistasis coefficients that you might get from a regression model, such as here, where you have this joint effect, to locus to locus or gene to gene, depending on the data set we're looking at, interactions that are directional. So immediately you can see you can't get this just from this phenotype X here, right? Because I have two parameters here, simply stated, and only one here that characterize the epistasis. But if I bring in another trait, then you can see that I've balanced out my number of parameters and also the information available. And so then I can do this transformation. And essentially what we're doing is taking the epistasis and we're rotating it in this phenotype space so that now it's aligned with the marginal effects from the two loci. And then now we can model this in terms of locus to locus effects. Okay, so it's a directed network we get of epistasis, and it's basically through reparametrizing regression coefficients for two or more phenotypes. Okay, so we wanted to look at this in as large of a system as we could find, and so we found what we call the little cross. It gives us a big network because it's an intercross of over 2,000 mice, but we call it the little cross because this cross is fixed for what's called the little mutation, which is a growth hormone receptor loss of function mutation that arose spontaneously. And so it's on both these backgrounds. And what that does is it allows us to study both bone and body mass independent of IGF-1 for the most part. So this is not an IGF-1 mutation per se. It's a receptor mutation that strongly reduces IGF-1 levels circulating. Okay, so we had that mutation fixed. Otherwise, we're looking at a black 6 C3H mouse intercross, F2, over 1,000 males and females. We measured a series of body composition traits. We genotyped. And we also looked at residual serum IGF-1. A few of these mice do have some IGF-1, and so that turned out to be interesting as well, as I'll get into. Okay, so we can do standard QTL scans on these mice, and you can see we get a nice pattern of QTL for these various traits. We scanned separately for males and females, and so you can see that there appear to be perhaps some sex-specific effects, like on chromosome 6 here for body weight, as well as some shared QTL across these traits. For instance, on chromosome 1, it looks like we have overlapping regions, etc. So we have a nice rich spectrum of QTL. It's a fairly well-powered study, especially for mouse intercross. And so we wanted to ask, well, what if we start to recombine these phenotypes into, say, principal components? So this is essentially what we're doing here. We're redefining phenotypes in terms of what we call eigentraits, and so you can see the first eigentrait is basically mouse size, the second is pulling in information on femur density, etc. However, when we scan these eigentraits, you still see this pattern of sort of unique and shared QTL. So it appears to be, to some degree, a pleiotropic system still, even when we do this transformation. And so this is a very nice substrate for this sort of interaction modeling we're doing. And so we went ahead and modeled the population, and here are the results. First of all, here are the QTL at each locus. So this is a summary scan, basically each column for the different phenotypes. Blue is low, brownish gold is high. You can see we have about 32 QTL. Many of them are shared across traits, although the effects are not always shared. Some are positive and negative. Sex and IGF-1, residual IGF-1, of course, have an effect on these traits as well. We then did our pairwise scans, and so here is basically an adjacency matrix of those interactions, if you want to think of it that way, or we have a source locus, a target locus. You can see we have these scattered interactions throughout the system here, 97 total interactions. So quite a bit of epistasis seen in this intercross. First, we looked at the QTL covariate interactions, that is to say sex by QTL and sex by IGF-1 interactions, and saw a couple of interesting patterns. So the first thing we saw in terms of sex, so this is encoded for the male sex, we saw that all the QTL interactions with sex were all positive. So these are all enhancing interactions, and the vast majority were these QTL two-sex interactions. So basically what this model is saying is that each of these QTL is enhancing the effect that sex has on all of the traits. Okay, so this surprised us a little bit. I was expecting we'd see sex modifying QTL that affect a certain trait like bone mineral density. It's the other way around. We're seeing QTL that seem to sort of affect everything by modifying the sex effect. And so that suggests that perhaps we should be looking for endocrine regulators within these QTL, not something specific to any given trait, but specific to the sex. For IGF-1, so this again is residual serum IGF-1, it's not the gene itself. We saw a different pattern, but a systematic pattern again, where we saw the enhancing interactions for the most part were QTL enhancing the effect of IGF-1, whereas the suppressive interactions, the negative interactions, were all from IGF-1 on these other QTL. And so our hypothesis is that we're looking at two different sets of genes here underlying these QTL. These ones that are enhancing IGF-1 effects, we think of as regulators or targets of IGF-1. That is to say, genes that harbor these variants are probably sitting in the IGF-1 pathway or interacting with IGF-1 in some way. So when IGF-1 is there, they can enhance that effect. Whereas these over here are sort of being suppressed by IGF-1. So these are basically variants that are only relevant when there's no IGF-1 in the system. So that you can think of this again as cryptic genetic variation, where in the absence of IGF-1, these pathways, or at least these genes, become relevant. And so that's why we think of these as the compensators for IGF-1, or maybe another pathway that you only see when IGF-1 is not there. So these interactions, both at the level of sex and with the serum marker, are giving us possible interpretations for these groups of QTL. We're also seeing sort of systematic effects too that we didn't necessarily expect to see. Okay, these can then help us think about models for candidate genes. And so our favorite candidate gene in this chromosome 10 QTL is Kitl, and that was mentioned the other night. That's the blonde gene. It also plays a role in a number of growth processes. And so here we had a interaction in which we have this QTL that harbors Kitl enhancing the effect of IGF-1, and they have opposite effects on femoral density. When we look at some existing data out there, we can see that Kitl has lower expression in the C3H strain relative to the black 6, so these are the parental strains of the cross. So we speculate that this is a loss of function, a loss of activity for Kitl in the C3H allele. And when you look at expression levels of Kitl and IGF-1, the gene itself, in those parents, you can see there's a negative correlation. Okay, so that leads to this model over here, which is consistent with our genetic model we got from the cross, which is that the C3H allele of Kitl reduces the activity of Kitl protein. Kitl protein is a suppressor of IGF-1 activity, and so and they both lead to increased femoral density. So when you lose the Kitl function, whatever IGF-1 is there is going to have a greater effect than it would have had otherwise because you lose that suppression, and so that's consistent with this genetic model. Okay, so this is an example of how we can better identify candidate genes and mechanisms through these interaction analyses. But back to the large-scale network. So when we look at the QTL-QTL interactions, there are 97 of these, there are quite a few. And so we wanted to ask, what if we partition them by patterns? And so we sort of came up with 4 different classes. We can have enhancing interactions with either the same marginal effects, coherent, or different marginal effects, which we call incoherent, and the same for the negative genetic interactions, or the suppressing interactions. And each of these have sort of a standard interpretation. These first coherent and enhancing interactions tend to give you more of the phenotype than either of the single perturbations or single variants, and so that's a synergistic interaction. These incoherent enhancing interactions tend to be sort of stabilizing. They look like this, where you have opposite effects for the 2 variants taken individually. You put them together, additivity would be up here, and they tend to push back down toward sort of the parental averages, basically. So that's a stabilizing phenotype. It's taking these extreme phenotypes and sort of combinatorially reducing that variance. We see a lot of redundant interactions, and these also reduce variance in the sense that you have two QTL with basically the same effect. You put them together, and you get a less than additive effect. And finally, we have these masking QTL. This is sort of like a classic molecular epistasis where you have opposite effect, marginal action that tends to express one or the other when you look at the epistasis, the pairwise effect. Okay, so when we look across all of our interactions, across all of our phenotypes, these are the statistics. And so, first of all, the synergistic, we don't see a lot of these. We see about 17% of our interactions, so that's statistically depleted, and you might expect that. They shouldn't be all that common. We do see some, but they're not terribly common. Stabilizing actually were the most common interaction we saw. There's a lot of stabilizing epistasis, at least in this cross. It's the majority or a plurality of the genetic interactions. A lot of redundant epistasis as well, and we did expect this because you often see this. You're perturbing different effectors in the same pathway, for instance, and you get less than additivity in the epistasis. And finally, we saw a few cases of masking epistasis, but this turned out to be the most rare in our population. Okay, so what does this all mean? Why do we have so many stabilizing interactions and redundant interactions? And so we came up with this fairly simple model of what may be going on. Say you have a trait like femoral circumference, one of them that we measured, and you have two different networks in the parents, and black 6 is sort of the wild type mouse in the mouse business, and so I'll say that's the normal mouse with the normal allele effects. And then C3H has a number of positive and negative effectors at a single variant level throughout that network. And so that's what you see when you look at the marginal effects. You're inferring these single locus effects, but when you start to pull out pairs of them, which is what you're doing in epistasis analysis, you start to bring in, say, a negative effector and a positive effector, and you put them together, and you're sort of balancing those effects out, and the epistasis you see sort of pushes that balance. And so here you can see the single effects sort of give you these wider phenotypes, and the combinatorial gives you something that's actually close to additivity, but is bumped down a little closer to the mean of the population. Whereas the redundant effects are pretty easy to understand in this context. You have a couple of negative effectors, but they're acting in the same pathway or in the same process, so when you put those two together, you don't get the double negative effect. Okay, and one more thing I wanted to note, and I just mentioned this basically, is that the stabilizing epistasis is not terribly statistically strong. In other words, we needed a very large population to even see it. And this is one way to quantify that. We're just looking at how much variance we fit for these traits in terms of the main effects versus the interactions. And so this often comes up, especially among studies of human genetics or medical genetics, where you're wondering how much of the variance is due to epistasis. In this case, even when we have a lot of QTL, not a lot of the variance is being explained by epistasis. It's mostly in the marginal effects of the QTL. So that gives us basically two conclusions, if I may extrapolate from an F2 intercross, which is, first of all, the majority of the genetic interactions are statistically weak, and or they're really phenotypically uninteresting. That is to say, they're pushing you toward the mean phenotype of the greater population. They're not giving you an extreme trait. Okay? And that may be sort of, frustrating if you're really interested in how important epistasis might be in some cases. But on the other hand, the interactions have provided us information on the architecture as well as how to interpret some of the interactions we see at the QTL. Okay, so I'll finish there. Primary acknowledgement goes to Anna Tyler, who's a scientist in my group who did the most of this work. We also collaborated with the Churchill and Donahue groups. And there's my funding, there's my contact. Like everyone else, I'm seeking postdocs, and there's an advertisement. Thank you.